Hi, I'm Pedro Sampaio, and I am the CEO and co-founder of Neobe Therapeutics. And I really appreciate this opportunity to tell you about the work that we're doing at Neobe, genetically engineering bacteria to overcome immunologically cold tumors. Now, we're doing this in the context of immunotherapies, which have revolutionized the clinical management of cancer by activating an immune response against cancer cells, eliminating them, leading to really strong, stable responses. However, still only about 15 to 20% of cancer patients respond to these therapies. And this is because there's a subset of patients that build a wall around them that prevents the efficacy of immunotherapies and immune cells to come into the tumor that is generated by their local microenvironment. Now, this microenvironment is a complex network that is made up of a number of cellular and acellular components that affect the way that a tumor grows and the way that it responds to therapy. It includes things like the vasculature, like components of the immune cells, and the extracellular matrix, a complex networks of fibers that provide structure to the tissue and that is intimately related to how tumors respond to immunotherapies. The extracellular matrix can create a barrier that prevents immunotherapeutic drugs to get into their site of action and do their job. They create a hypoxic and low nutrient environment that can affect the response to immunotherapies. But more importantly, the extracellular matrix can create a barrier to the infiltration of immune cells, trapping them and preventing them from getting to cancer cells and doing their job. This leads to what is known as immune-excluded tumors, something I saw myself working as a postdoc at the MD Anderson Cancer Center in Texas, where we analyzed the spatial distribution of different components of these microenvironments, and we saw that cancer reactive CD8 positive T cells, particularly in pancreatic patients, tend to localize within these interstitial stromal regions and not close to the cancer cells. And these areas where T cells localize tend to correlate with high levels of extracellular matrix deposition, like collagens, for example. So if we know that these extracellular matrix components prevent immune cell infiltration into the tumor, then why are we not doing something about it? Well, it turns out that we are. There's a lot of data that shows in animal models of cancer that if you disrupt the extracellular matrix, you can increase the immune infiltration into the tumors. And on the left here is one example using pancreatic cancer genetically engineered mouse models. And by using a drug that disrupts the cross-linking between collagen fibers, you see in the bottom here that you suddenly have a lot more immune cells getting into the tumor than you do in the controls on the top. However, when these approaches were taken to the clinic, you saw mostly a response like you see here with a drug that was put into the clinic by Halozyme that degrades hyaluronic acid in the tumors, and that didn't really lead to much of a response at all. And there were two main reasons that ended up accounting for this. One is accessibility. Biologics like antibodies and enzymes tend to struggle to go past these fibrotic barriers and do their job. But mostly it's toxicity because the components of the extracellular matrix are common to the healthy tissues. So systemic delivery of these drugs tend to lead to really severe toxicities, and the doses have to be brought down so much that they end up just not really doing anything to the tumor. So when you take a first principles approach to look at this, it becomes clear that what we need is a brand new solution that allows you to disrupt the extracellular matrix locally without leading to these problems. And that's the solution that we believe we have at Neobe, where we're engineering safe tumor colonizing bacteria to degrade these extracellular barriers to infiltration and enable immunotherapies in non-responders. There's a number of reasons why we think bacteria are the right way to do this. They have an innate capability of colonizing tumors, and they have molecules on their surface that can trigger an immune response. But most importantly, we have the genetically engineering tools, the synthetic biology tools, to engineer them to produce a payload under specific conditions. And by doing this, we are developing our technology at Neobe, where we're engineering tumor colonizing bacteria to be activated by the physiological conditions within the tumor to produce a payload that will degrade extracellular matrix components only in the tumor, enabling immune cells to enter the tumor and these patients to now respond to immunotherapies. We're doing this in the context of a growing field of bacterial therapeutics in cancer, and it is the right time to use these tools to target the microenvironment. All other companies using bacteria for cancer are either using them to directly target the immune cells or directly kill cancer cells. No other company is using this technology to disrupt the extracellular matrix, which puts us in a pretty good position to address immune-excluded tumors. Our vision is to create a company to create a portfolio of bacterial components to target different components of the microenvironment, going from the product that we're engineering now to a plethora of products targeting different components of the stroma to facilitate therapeutic efficacy, and eventually develop into products that can be used both as microenvironment modulators and immunotherapies. We've got a great team to be able to achieve this that combines cancer biology and bacterial engineering expertise in my co-founder Annalise, who's been working in that field for several years. And we're lucky to have the support of a great board of advisors and Deep Science Ventures and Cancer Research UK to be able to achieve this. If you want to find out more, either go to our website, connect with us on LinkedIn, or you can find my co-founder Annalise, who will be in person at Genesis and will be happy to take your questions. Thank you.